Welcome back to the Genetics Lecture Series. In this podcast, we're going to learn about spinal muscular atrophy, the most recent condition to be added to the newborn screening panel in Alberta. The objectives for today's podcast are to review the genetics of spinal muscular atrophy, to review the spectrum of phenotypes of SMA, and to discuss how understanding the genetics of the condition has informed new therapies for SMA. Spinal muscular atrophy is an autosomal recessive condition characterized by loss of lower motor neurons resulting in progressive proximal muscle weakness and atrophy. The gene that is implicated in SMA is called survival motor neuron 1 gene, which we call SMN1. It is located on chromosome 5 and codes for the SMN protein, which is thought to have a role in mRNA synthesis and inhibition of apoptosis. All forms of spinal muscular atrophy are due either to deletions of exon 7 on both alleles of the gene, or one deletion of exon 7 and one mutation, usually a single nucleotide variant, resulting in deficiency of the SMN protein. There are 5 types of SMA which vary by age of onset and severity of symptoms. Type 1 is the most common, affecting approximately 45% of patients with spinal muscular atrophy. Infants affected with type 1 have no symptoms at birth, but by 6 months of age present with proximal muscle weakness, respiratory insufficiency, and poor feeding. Patients with type 1 usually do not achieve the ability to sit, and the majority die before the age of 2. On the other hand, patients with spinal muscular atrophy type 4 will only notice mild symptoms later on in life, if at all, and their life will not be limited by the condition. If all patients with SMA are for the most part homozygous for deletion of exon 7 on SMN1, how can there be so much variation in the phenotypes between them? This perplexed scientists and clinicians for quite some time, until we learned that there are actually 2 genes that produce the SMN protein, SMN1 and SMN2. The difference between these 2 genes is a single nucleotide variant found in exon 7. In the SMN2 gene, there is a thymine in the place of a cytosine. This variant causes the splicing out of exon 7 in about 90% of the mRNA made by SMN2. As a result, the wild-type version of this gene makes a truncated and for the most part non-functional protein 90% of the time. About 10% of the time, however, exon 7 is included in the mRNA transcript, resulting in a full-length, fully functional SMN protein. So, someone with spinal muscular atrophy is not able to make SMN protein from their SMN1 gene, but they are able to make some protein from their SMN2 gene. To make things even more confusing, the 10% of functional protein made by SMN2 is not actually 10%, because SMN2 exhibits copy number variation. In this case, the coding sequence of SMN2 can repeat itself, with the final number of copies varying from person to person. In the general population, studies have shown that people without spinal muscular atrophy have between 0 and 8 copies of SMN2 in their genome. Patients with SMA have at least 1 copy of the SMN2 gene, but what is notable is that their phenotype correlates to the copy number of SMN2. The majority of people with spinal muscular atrophy type 1 have 1 to 2 copies of SMN2, with patients with type 3 or 4 having 4 or more copies of SMN2. As mentioned, spinal muscular atrophy is the most recent condition being screened for in Alberta newborns. And this is in large part due to the advocacy efforts of families affected by spinal muscular atrophy, as well as new treatments. There are 3 treatments for SMA that have been approved in Canada. 2 target endogenous SMN2 with the goal to increase SMN protein production, and 1 introduces new SMN1 into the patient. Nusinersen and risdiplam promote the inclusion of exon 7 in mRNA transcripts of SMN2, resulting in a greater number of fully functional SMN proteins being formed. With nusinersen, an oligonucleotide that binds intron 7 and prevents other splice factors from splicing out exon 7, patients receive 4 injections over 2 months, then 1 injection every 4 months. Risdiplam, a small molecule that binds pre-mRNA to stabilize protein production resulting in inclusion of exon 7, is a daily oral medication. Zolgensma is a one-time gene therapy infusion that delivers a new SMN gene to the motor neuron cells all over the body using a viral vector. Unlike the SMN2 targeted treatments, which attempt to manipulate the genes already in the body to increase SMN production, Zolgensma introduces a new gene which is taken up by the cells and is engineered to continuously work in the body, producing SMN protein. So we have a rare condition that is treatable. This is exactly the kind of condition that is completely appropriate for newborn screening programs. In a pilot project between 2022 and 2023, 47,005 samples were analyzed for SMA in Alberta. Of these, 6 screened positive and 5 went on to have diagnostic confirmation of the condition. Treatment was started in 4 of the infants between 25 and 30 days after birth, and the 5th started treatment at 71 days of life after developing areflexia and sparse tongue fasciculations. In Alberta, all 3 treatments for spinal muscular atrophy are fully covered. Caregivers and physicians work together to choose the best treatment option for the patient, recognizing that each treatment has its associated side effects. After the pilot program was complete, spinal muscular atrophy was officially added to the newborn screening programs in Alberta. It will be interesting to see how advances in technology, drug discovery, and our understanding of variants in the genome change the newborn screening landscape over the next few decades. It is quite likely that before you're done residency, the panel in Alberta will look a little bit different than it does today.